Good afternoon, everyone. My name is Caitlin Raring, and I am a research chemist at the USDA. And it is my honor today to be hosting the last, I believe, ACS webinar or agro webinar of the year, presented by Professor Adam Wong. So Adam joined the University of Florida Entomology and Nematology Department in 2017. Prior to that, he served as a postdoc at the University of Sydney and Harvard Medical School. And he received his PhD from Cornell in entomology in 2013. So his current research is focused on the insect microbiome, and particularly the nutritional and chemical basis of host microbe interactions. So in just a moment, I'm going to turn it over to Professor Wong. I do want to point out that we're going to hold a quick discussion at the end. So as you're listening, please feel free to type any questions or research topics you want to bring up in the question box located in your webinar panel. And I will read those to the audience. So with that, I want to thank Adam for taking the time to share his research with us. And I'll turn it over to you, Professor Wong.
Okay, and thank you very much, Caitlin, for having me here, and thank you, ACS, for having me as a webinar speaker. I have never done something like that before. I always attend a meeting in person and talk in front of people. So this is something for a change, and I'm really excited to be here. So today I'm going to talk about my research program using knowledge from insect microbiome interactions and applying it into new management tactics for invasive fruit flies. And this is an area that has been going on at least for the past 2 years since I joined the University of Florida. And I believe many of you know that Florida is a hotspot for pest problems, and we have a lot of agricultural needs to tackle many insect problems. So just to give you a little bit of ideas on the big picture on why we're interested in invasive fruit flies. If you think about the global market value for fruit and vegetable productions is a significant part of the human world. Currently, the market value is estimated at $240 billion in value, and it's projected to grow very well at 7% per year, meaning that in the next 5 to 10 years, it's going to reach $400 billion mark. So it's important for the economy that we sustain a productive system for making and producing fruit and vegetables. And in the developing world, fruit and vegetables are also a cornerstone in solving many of the issues associated with poverty, hunger, and malnutrition. And to keep pace with fast-growing demand on fruit and vegetables, as we all know, we all love our fruit and vegetables. We want to eat healthier, and we all want to have good nutrition in our diet. One of the biggest challenges to maintain this agricultural productivity is crop losses due to insect pests. And particularly, the fruit fly is a big problem for fruit and vegetables productions. As many of you have heard, there are many different fruit flies that can attack crops. There are two main families, the Tephritic fruit flies and Drosophila fruit flies. Together, there are over 8,000 to 9,000 species of fruit flies, and at least 250 of them are significant pests that can cause economic losses. So my program is to focus on fruit flies, and I can tell you that even though currently I'm working in entomology department, trying to do some more applied work looking at fruit fly microbiome, I wasn't trained with that background. I started off as a basic scientist studying the microbiome in Drosophila melanogaster, which is a great model system to understand host microbiome interactions. And gradually, I've become more interested in solving some of the real-world issues. And one of them is the Drosophila suzuki highlighted here, which I'm going to talk about today. And this pest is highly invasive, and they can attack a range of soft-skinned fruit, and they are causing billions of dollars in agricultural loss in the world, and as at least a few $100 million losses in the US. I'm also collaborating actively with researchers from around the world, looking at different fruit flies that are attacking our crops and vegetables. For example, I have some colleagues working on the mayfly, Mediterranean fruit fly, one of the most serious crop pests. And my collaborators from Israel and I actually just won a grant recently looking at the microbiome of this pest and trying to develop microbial-based tactic to control it. I also have collaboration with people from Australia, which are devastated by the Queensland fruit fly, Bacillus trioni. We are also developing some microbial-based method to trap this particular fruit fly pest. I also have some collaboration with people in Florida and also other parts in the US looking at oriental fruit flies. I think some of you might already heard there was news about the oriental fruit fly causing outbreaks in Florida. Last time when it happened, it was back in 2015, almost 3.5 years ago. And it devastated the whole vegetable and fruit productions because this pest can attack over 400 different crops, and it cost the Florida over $30 million to eradicate it, and a lot of small grower out of business. So we need to pay attention to some of these fruit fly pests, because once they invaded a new area, it can cause damage very quickly, and by the time you know it and start making some of the measure to control it, it becomes a serious cause and problems. So for today's talk, I'm mostly going to start talking about the basic research models of Drosophila melanogaster, and then we gradually switch to the spotted wing Drosophila as a pest. I'm going to talk about 3 different areas on how microbiome affect the pests or the fly in general. I'm going to talk about development and nutrition a little bit, and then I will move on to talk about how the microbiome are modulating the behavior and the olfactions of the flies. And finally, how I can extend and invented new knowledge and apply into pest management program. So to give you a quick overview, just some idea about microbiome fruit flies, we have been working on Drosophila melanogaster for nearly 10 years. And we learned that the microbes carried by a fly is actually quite dominating. There are roughly at least 100,000 to 1 million microbial cells that are present in a single fly. And these numbers are based on studies in the lab, so meaning that the number could potentially be even higher in the flies in the field, because number of studies show that the flies in the field carry more microbes. Many of these microbes are bacteria, but they also carry some fungi, and many of them are located in the gut of the flies. We have sequenced the genomes of many of these microbes, and the size of the genomes are somewhere in the range of 3 megabase size or bigger. So collectively, we're talking about almost 300,000 to 3 million megabase of genetic material that are coming from microbiome presence in the fly. And you think about the genome size of the fly, which is only around 175 to 180 megabase pair, we're talking about almost 1,700 times more genetic material coming from the microbes in the fly, in contrast to genetic material coming from the cells of the flies. So it's a remarkable contrast if you think about how microbes and all the genetic activity that can come from microbes that influence the flies. And we know that they do affect the flies because we have studied it now and have a lot of evidence. And this is just a fun way to illustrate the scale of contrast. It's kind of the size of the Statue of Liberty versus the size of a human being, which obviously you won't be able to see in this picture. In terms of the composition of the Drosophila microbiome, we know that oftentimes they are dominated by 2 groups of bacteria, acidic acid bacteria such as Acidobacter and Gluconobacter, but also lactic acid bacteria. These 2 groups of bacteria are also pretty big groups, and They do all kinds of metabolic activities such as fermentation or oxidations of different sugars and ethanols, and some of them can also synthesize amino acids and vitamins and other micronutrients. We know that these microbes can be passaged from mother to offspring because when the parental flies lay eggs, they will deposit on the egg surface some of the fecal material that are microbially rich. And when the newly hatched larvae consume the eggshell, they become contaminated. And that's one of the principal routes that the Drosophila can pass the microbiome from one generation to another. And these bacteria make up almost more than 95% of the microbiome, based on study from the lab, and their presence across laboratory Drosophila as well as in the field as well. So they're widely present, they are very abundant, and they are easy to culture. And one of the nice things that we can use Drosophila to study microbiome functions is that the microbiome can be manipulated experimentally, and it's pretty easy to do. So as I mentioned, the microbes are passaged from the parents to offspring through egg deposition. So if we remove the microbiome deposit on the eggs and then place those eggs into a sterile environment, we can generate a germ-free or acidic flies, and we can also reassociate the association of the fly with the microbes by feeding them back with some of these microbes. So we can generate gnotobiotic flies. And this system is very powerful because we can selectively reassociate certain interactions and study how that affect the fly performance and different phenotypes. So to give you one example of the study that has done more than 5 years ago now. So we set up this experiment looking at how the microbiome affected the nutrition of the flies by placing conventional eggs, which has the microbiome, and the acidic eggs, which doesn't, into a diet treatments. And there are in total 16 different diets being set up. So the diet made of yeast and sugar. So yeast provide the source of proteins, but also different macronutrients, while glucose is the carbon source. So we set up different concentration of yeast and sugar so that we have different total quantity of nutrients, but also relative of nutrients. As you can see from the results here, we measure the larval development of the conventional and acidic flies. The black is the conventional larvae and the red is acidic larvae. You can see here that when there is plenty of yeast, meaning that there are plenty of proteins and macronutrients, the development of conventional and acidic flies are pretty similar. They took around 11 to 12 days to develop from eggs to adults. However, if you put them into the condition where the yeast become very limited, such as 25 grams per liter versus 200 grams per liter sugar, you have a different pattern, right? You can see that the acidic eggs fail to develop, or they develop much poorly, or they have a much prolonged development time. And what that means is that the microbiome is important for host nutrition, and that is important for larval development, but only when the diet has a limiting yeast relative to sugar. And this diet shown here, I believe, are more realistic in nature, because if you think about it, right, a lot of these flies are feeding on fruit and vegetables, and fruit and vegetables usually do not have a rich source of proteins or amino acids or even some of the vitamins. So they are actually feeding on a high sugar diet in many cases. And so the microbiome potentially will help them to cope with what's in nature. You know, when they're feeding on strawberries or blueberries or things like that, then they would need the microbiome to help them to survive and develop properly. And we also did a little bit of genome sequencing to look at what are the metabolic basis on how microbiome support Drosophila growth and nutrition. We sequenced many of the genomes from the bacteria isolated from Drosophila, and we also have the data from fly genome. So we were able to reconstruct the molecular basis of nutritional interaction. So here's one example of the prediction we got by looking at the genome data. And we found that many of the enzymes, genes that encode those enzymes, involved in B vitamin biosynthesis, such as riboflavin, one of the most important B vitamins, are present in the genomes of the gut bacteria in the flies. Many of them are absent in the fly genome. So you can predict that the pathway that synthesizes riboflavin coming from a precursor with the GTP or the ribulose 5-phosphate are all mediated by the bacteria. Some of the enzymes, on the other hand, are shared between the host and the microbiome, so they're present in both. So you can predict that the riboflavin can be kind of transported into the host and be converted in some of these intermediate compounds or cofactors used in respiration and growth and development. So we predict, based on the genome data, that riboflavin synthesis is mediated by the microbiome, and we were able to also test that by doing a dietary supplementation experiment where we supplement the diet of acidic eggs on the high sugar, low yeast diet where they don't develop with B vitamins. We give them a mixture of 8 different B vitamins. And as you can see here, with those supplements, there's no surviving flies. If you give them supplements, then the acidic flies start to develop much better, although still not to the full extent, but it massively rescue the development. We also selectively delete some of the B vitamins and found that the riboflavin, which becomes a top hit in our genome data, happens to be the very important B vitamin for development. So if you delete riboflavin from the mixture, then they don't develop very well. So by doing a dietary supplementation experiment, we were able to validate some of the genomic data. So now I'm going to move on to another example on how we use the nontobiotic approach to study microbiome impacts on Drosophila. And this is one of the very exciting area because we start to find that the microbiome can modulate the behavior of the flies. Now, before we start that research, there are already a body of literature that show Drosophila flies, the behavior of them are modulated by microbes. For example, there are papers that show that the fly can be attracted to yeast, which is one of the most well-established fact that fly can seek out to yeast and they would like to feed on yeast. And they also identify the volatile compounds that are driving the attractions and the genetic basis on these volatile productions by the yeast. Study also identify the mechanism by which the Drosophila can avoid certain microbes. So some microbes, particularly fungi, are harmful to flies, and the fly have a sensing mechanism by olfactory receptors to avoid these harmful microbes. There are also some studies that suggest Drosophila, if they do not constantly feed on beneficial microbes, particularly bacteria, they will lose it eventually. So that motivates them to constantly seek out these beneficial microbes. So because of those body of work, we asked the question, how do the flies kind of sense and seek out beneficial microbes in the environment? And can the symbiotic microbiome modify or affect the host olfactions and behavior? To test those questions, we set up this 4-agent assays. So in these assays, we have Drosophila fly that has not been fed for a while, so that they don't have any food in their stomach. And then we place them into a arena with different food choices. So we have food choices that are made of just a protein sugar source, and then we contaminate it with different microbial isolate. And these microbial isolates, such as Acidobacter or Lactobacillus, are found from the fly naturally. We also present them with a pathogen, Staphylococcus species. And at the same time, we set up notobiotic flies, so the flies have different microbiome composition to start with. And then we monitor how the fly will make food choices and pick up those food. And from our results, we found something very interesting. First of all, we found that the conventional fly, so the fly that has the full microbiome, are strongly attracted to food that has been seeded with acidobatters. And as you remember, acidobatter is a dominant bacteria in the fly gut. So it's perhaps not very surprising that the fly has a strong attraction towards this bacteria. They also have a moderate um, attractiveness towards different lactobacillus. And that also makes sense because lactobacillus also is a member of the fly microbiome. And they certainly have less attractiveness towards the food that has no microbial contamination or towards the pathogens. So what that tells us is that some of this fly-associated bacteria could be potentially very helpful in developing into attractant for these flies. What's more interesting is that we found that when we put the flies and raise them germ-free, so when they do not have microbiome or have any history of microbial exposure, then one of the acidobatter, particularly the dominant one, the Acidobatter pomorum, become not so attractive. And the attraction towards Acidobatter tropicalis also reduced. So what that suggests is that the microbiome or the history of the exposure really plays a role in modulating the attractions towards Acidobatter. But interestingly, they still retain a very strong attraction towards Lactobacillus. And what that suggests is that the fly has an innate attraction response towards lactobacillus, and they have a higher tendency to feed on food that has no bacteria when they has been raised in a germ-free environment. And by feeding them with Acidobatter or lactobacillus through their development, you can see they have reinforced the attraction. So the Acidobatter associated flies have a strong attraction towards Acidobatter. Likewise, the Lactobacillus associated fly has a strong attraction to Lactobacillus. And we also test this kind of phenotype, very surprising results in larvae. So just to give you an idea, most of the time when the fly lay eggs, they deposit a lot of microbes on the egg surface, but predominantly the Acidobatter and Lactobacillus. So we set up these larval choice assays. We place the eggs in the center of a petri dish with agar, and then we give them to 3 different food choices, either food that has not been contaminated with any bacteria, or food that has lactobacillus, or food that has Acidobatter. And what we found is that in the conventional eggs, which means the eggs that has been contaminated with the microbiome, the larvae, the newly hatched larvae, will quickly seek out food that has Acidobatter and lactobacillus. So the germ-free eggs, however, do not have the same phenotype, behavior output. You can see that they kind of more randomly go over and seek out different food, suggesting that the microbiome in the early exposure on the eggs and larvae really plays a role in modulating how they perceive Acidobatter and lactobacillus. And you can see even after 2 days, they start to seek out a little bit more on Acidobatter or lactobacillus. The magnitude of attraction is still much weaker compared to other treatments. So that suggests the microbiome does play a role in modulating the olfactory guide preference towards microbes of the flies. And now my students and I have done a little bit of testing looking at the molecular mechanism that drives Drosophila attraction towards Acidobatter and lactobacillus. So one of the things we can do with Drosophila melanogaster is that we can set up a genetic knockdown using a GAL4 UAS system. So the idea is GAL4 is like a driver that is attached to any tissue-specific gene of your interest. So particularly, we have an Invasive driver that's only specific in the brain, and we use that GAL4 driver to knock down the olfactory receptors in the flies. And there are roughly 67, actually more than 67, it's like 70 olfactory receptor in Drosophila. So we were able to knock down those receptors and see how by knocking down those receptors, would the fly change the behavioral tendency towards these microbes. So we set up over 1,000 of these traps testing Acidobatter and lactobacillus. and we knock down every one of these olfactory receptors and see how they become trapped in these assays. So here is just a figure showing you what the set was like. So we have the fly put in the cage with the 2 cups. So one of them contain the Acetobacter with the medium, and the other is just a control without the Acetobacter, without the lactobacillus. And we were able to observe how the fly will enter the trap with the Acetobacter or lactobacillus or without. And over the screen, we identify a number of receptors. As predict, a lot of receptor has absolutely nothing to do with the attractions. So by knocking down those receptors, the olfactory receptors, the attractions of the flies towards Acetobacter or lactobacillus have not changed. But we do find a couple that seems to be important. So we now have a few candidates. I think about 5 of the olfactory receptors that we knock down, and by knocking down those receptors, the fly become much aversing the Acetobacter or lactobacillus. So for part 1, it's all basic science, and it described over 10 years of work that we work on Drosophila melanogaster. We learned that the microbiome supports larval development, particularly on diets with low yeast and high sugar. And we also, based on the genomic data and dietary supplementation experiment, we know that the microbiome helps the flies develop by providing B vitamins, particularly riboflavin. We also have some new emerging data that suggests the microbiome can affect the microbial preference of the flies, and also ultimately how they forage, the behavior on how they make food choices. So now I'm going to move to the second part of the talk, which now goes to the more applied side of things, talking about Drosophila susukii, also known as the SWD spotted wing Drosophila. So SWD, I think many of you probably know, is an invasive pest and is a significant economic problem to agriculture. To give you some idea about the biology, this pest takes about 7 to 12 days to develop from eggs to adults. A single female can produce over 200 to 600 eggs in her lifetime, and they tend to live somewhere between 3 to 9 weeks. And one of the unique features about SWD is that they have a serrated ovipositor that can piece open an intact fruit and then lay the egg inside the fruit. So many of the other Drosophila species tends to be kind of scavenger, and they tend to feed on damaged or rotten fruit. But SWD is different. They attack intact, healthy, ripening fruit. And as a result of a very highly reproductive cycle and short generation time, SWD has been causing millions of dollars in agricultural losses in small fruit in the US. And one of the challenges during SWD is that they can attack many different crops. They are not specialists. They can attack a varieties of fruit as long as this ovipositor can kind of attack and piece it open. So it has been a big problem, particularly on strawberries and blueberries in Florida, but they can also attack cherries, raspberry, peaches, grapes, and other fruits and vegetables. And if you look at the invasion of the pest, it's a huge problem. So here you can see in the map here, SWD was first invaded US back in 2008. It came from the Southeast Asia and was detected in California for the first time back then. And then in a few years' time, it started to spread to many different states, including Florida, in just 1 year. And now it's pretty much detected all over the place. This is already an outdated map in 2013, but right now it's detected in over 42 different states in the US. And taking Florida as an example, it was detected in one county, Hillsborough, in 2009, and then in just 4 years, it has spread all over the place in Florida. It's highly invasive. In just 4 years, it's now become a big problem for crops, and it's causing millions of dollars losses. So managing SWD is a problem. is still a big challenge. And there are many reasons for that. For example, if you think about intervention, without any chemical insecticide usage or sanitation effort, the pest can destroy the crops. Total loss, almost 80 to 100% loss as a result of SWD. In many of the fresh fruit markets, they have a zero tolerance policy, meaning that if they can detect even a single larvae in the batch of fruit, they will reject the whole load. So that would cause a lot of losses for growers because they cannot market or sell those products. And as a result, a lot of growers and industry are very paranoid of having SWD, and so they are spraying a lot of the insecticides on the crops, even though sometimes to a level that's so excessive and costly. The other problem in insecticides is that they're not very effective against larvae, right? Because the female lay eggs inside the fruit, so the larvae tends to infest the insides of the fruit. So by spraying a lot of insecticides on the surface of the fruit doesn't really help much, unless it's really kind of at the early stage when the flies are just rare and very small in numbers, and you can kill them before they become established. And however, to do that, you need to have a good monitoring system, meaning that you can detect SWD when the population is still low, and the development of traps and loops that are selective efficient is still not there, and it becomes one of the number one priority agreed by many of the IPM experts. So there are many issues dealing with SWD. The management is still suboptimal, and we need to have a better strategy than constantly spraying chemical insecticides. So we asked the question, could we leverage some of the microbiome knowledge that we learned from Drosophila melanogaster and also extend some of the new ideas to better manage this pest? So one of the things we are pursuing now is to identify how and why and how the microbiome promote larval development for SWD. So one of my students, Sean, has been working on this project, looking at the specific bacteria or fungi in SWD that are important for the development. So he set up some simple assays using strawberry-based medium. So we just crush some strawberries, mix them in agar, and then make those media. And then we place conventional SWD eggs as well as acidic germ-free eggs onto the strawberries. And what we found was that the conventional egg, as expected, they develop very well on strawberries. So they have somewhat at 80% survivals. So they have a high survivals on strawberries. And that's quite incredible because strawberry is pretty much a pack of fructose and micronutrients. And the larvae can fully develop on those diets without any other nutritional need, meaning that the microbiome must be playing a role in supplying some of the nutrients that are lacking in strawberries. So in acidic eggs, they don't develop at all. We found absolutely no survivors. They just all die, and we barely see any visible larvae. So they must have died at a very early stage. So they cannot live on strawberries without their microbiome. And that's a good news because that means the microbiome could potentially be a target that we can eliminate or disrupt in order to disable the flies to develop on strawberries. We also isolate a range of microbes from the SWD gut, and we found that they can rescue if you feed if you supplement those microbes back in the food, they can rescue SW development to a different degree. So if you supplement them, the food with acidobacter, the dominant acidobacter that are found in Drosophila gut, it brings up the survivorship somewhat at almost 50%. It's still not to the full extent, but it does have a strong effect in rescuing the acidic eggs. But some other microbes we isolate, including 2 of the yeast species, surprisingly did not play a strong role in helping them develop. We were a little bit surprised because when we first did this experiment, we thought yeast is kind of the nutrient that can supply to the fly, and maybe that's how they can survive on strawberries. But to our surprise, the two yeast species does not rescue them to the level comparable to Acetobacter or conventional SWD. So now we're doing some transcriptome analysis, trying to figure out what are the genetic products derived by the microbiome that are important for development. And other avenues that we are pursuing now is looking at kind of discovering new fly attractant and repellents. As I mentioned earlier, there's still a lack of effective and specific attractant for SWD, and that makes monitoring and detection very difficult. So my student Jordan and my postdoc Masru are now looking into microbial-based attractants and repellents. And one of the observations we found that kind of leads to that direction was that we found that the microbiome composition of SWD, as shown here, are pretty much dominated by some of these acid bacteria, Acetobacter and Conobacter. There are many species of them, but in general, these two groups of bacteria dominate the SWD microbiome. We also look at the microbiome of a strawberry that has not been exposed or visited by the SWD. You can see that it's pretty diverse. It has all different genera of bacteria present. However, when a strawberry has been visited or foraged by the SWD, the microbiome has been completely altered as a result of SWD. So you can see that the foraged strawberry has a microbiome that seems to more resemble of in the SWD, and it seems to lose or become outdominated by these groups of bacteria, particularly Acetobacter. We also found that the foraged strawberry are much more attractive to SWD than unvisited strawberries. And what that means is when the SWD visited strawberries, the two has very different microbiome, but the SWD alter the microbiome of the strawberries. And as a result, the strawberry have a different microbiome, and they potentially have a different volatile makeup and become smells differently to the flies apparently. And the flies would prefer the foraged strawberries. And that might be a way that the fly can drive aggregation in the feeding towards the crops that they're targeting. So Jordan also spent a little bit of time isolating some of these microbes from strawberries and also from the flies, and developed a library of all the microbes. And by having those libraries, we were able to test the attractants or repellents towards SWD. So we can seed those microbes into these trap assays and have a control that has no microbes. And then we can test whether the fly would prefer the medium with the microbes or without. And the medium, we've been using a berry juice, but we are now moving to test strawberries or blueberries, the whole fruit itself. And by doing these trap assays, we can then test which of these microbes are attractants and which of these are repellents. And if any of these are neutral, doesn't affect the attractions or repellency of the fruit. And by using these assays, we already isolate a range of micro candidates. And you can see from this figure here, we have a few candidates that are driving a strong attractions. So the attractive index has improved significantly as a result of having those microbes. We also have a few microbes that seems to be repelling the flies. So if you add those microbes to the berry juice, the fly will avoid it. So that could be potentially a candidate for repellents. And right now I'm collaborating with the USDA chemistry unit, trying to figure out what are the volatiles coming out from these microbes that mediate attractions or repellency. So I'm just going to show you one example. So we pick up two of the microbes, Pentanor agromerans and also Phycobacterium fenae. So the PA is kind of like a candidate for attractants, and the FF is a candidate for repellents. It's interesting because PA is also known to be a competitor for plant pathogen. It's been used as a plant probiotics to fight some of the fungal diseases in apples and pear. So here's the latest results we got. So we have our collaborator John Beck and James Brown helping us to run some GCMS looking at the volatile profiles of these two candidate microbes. As you can see here, when we add those microbes to the fruit juice at the beginning, at 2 hours, the volatile profile of the juice are pretty similar, so they haven't really changed much yet. But over 24 hours, overnight, then you can see that the repellent microbes, the one in circle, FF, would change the volatile profile to here, and it's sustained that way after 48 hours. So this is the volatile profiles of the repellent microbes. The attractant microbes, on the other hand, has a very distinct volatile profiles. After 24 hours, they shift very differently. And then in 40 hours, it changed further. So now we have a list of volatile compounds based on the initial GCMS data that we think might be important for driving attractions or repellency to SWD. So with that, now I just want to summarize the different avenues that we are now developing, making use of the microbiome of the flies to develop strategies to control this pest. So we are now using some of the omics techniques, transcriptome and metabolomics, to identify how the microbiome enable SWD larvae to develop in the fruit, particularly strawberries. So by doing so, we can be able to develop some approach to disrupt those molecular targets, such as genes and proteins that are coming from the microbiome. We are also looking into the attractant microbes and repellent microbes. As I show you in the data, we have some candidate microbes that we think are potent attractants or repellents to SWD. And we are now testing more, and we're hoping to also test mixture of microbes. So far we only done single species. We're hoping to do a little bit more by looking at mixtures. And with that, we can develop a more effective monitoring system, more effective traps, and hopefully can also have a repellent that can protect the crops just by simply spraying the crops. And with that, I would like to finish and would like to acknowledge people in my lab, particularly Masru and Shao, who are the senior members supervising some of the undergraduate students, Sean, who take care of the SWD microbiome project looking at development, Jordan and Michael, who look at the attractants and repellency. And I would like to thank USDA for their long-standing support, particularly John and James, who has been absolutely amazing doing all the volatile work. Also collaborator in the department who are experts in blueberries and strawberries, and my collaborator from other countries in Australia and France and Israel, and all the funding support that has enabled the work. And with that, I will finish here, and I will be happy to take questions from the audience. Thank you.
Awesome. Thank you so much, Adam. That was a really interesting talk.
Thanks.
I want to remind everyone, please use the question box to pose any questions to Adam that you might have. In the meantime, I have quite a few questions for you. So you mentioned there at the very end that you're interested in pursuing mixtures of microorganisms.
Yes.
And I'm just wondering what the motivation behind that area of research is. Why are you looking at mixtures, or why are you curious about mixtures?
Well, that's a great question. So oftentimes when we are looking at metabolite productions in microbes, they often work in a community fashions. So if you have a single microbes, they might produce certain metabolite, but they're often not very potent. And also when you have mixture, they will produce something new as well. So there's a lot of synergy that tends to happen in mixture. So for example, let's say if we identify 3 or 4 different attractant microbes, and by mixing some of this together, you might get an even stronger response, even stronger attractants. And that's because certain metabolites are only produced through like a collaborative metabolism of multiple species. And we have seen that in some of the literature recently, that if you have multiple microbes mixed together, the potency or attraction or repellency is much greater than any of them individually. So by doing mixture, we can have a better chance of hitting metabolites or chemicals that can be even stronger attractant or repellents.
That's interesting. Is there any hypothesis or consensus out there about what drives that sort of non-additive attraction? Like,
Uh-huh.
do they people think it's volatile based, or is it seem to be more gustation based?
Right. The few paper that I read seems to be a lot of volatile base. So they quantify the volatiles in single species and in volatiles, in mixtures. And they found that in mixture, there are a list of volatiles that only present in mixture, and it's not present in any of the individual any individual species
Hmm.
culture. And then they test those volatile and found that they are very attractive to flies. There's a paper in eLife last year that show some of the mixture, not just mixture of bacteria species, but mixture of bacteria plus yeast are much more attractive than the bacteria or yeast alone. So it seems to be a lot of volatile bases in those difference in potency.
That's fascinating. Well, I'm interested to see what John and James come up with for that
Yeah.
in your collaborations. Cool. Um,
Yes.
Another question for you. So you spoke, the first part of your talk was devoted towards these really elegant laboratory-based studies with sterile flies. I'm wondering
Mhm.
about, if you were to go catch these, say, the SWDs in the
Yes.
wild,
Mhm.
what their gut bacteria might look like,
Yes.
and how that compares to the communities that you modeled in your systems.
Great. That's another great question. So there are a couple studies that look into the SWD collected from the wild, and they often found that they do have a high abundance of acetic acid bacteria, including Acidobacter and Conobacters,
Mhm.
so that they're pretty widespread in SWD. Now, the two studies that has looked into microbiome in wild SWD, they collect the flies from a berry crops orchard. So it's unclear that whether SWD in other environment might have different microbiome or there might be more variations. Um, there's also a study that look at diapause SWD, and interestingly, the diapause SWD seems to have a very different microbiome, and we still don't know why. Maybe they have a very different dietary habit at the time during overwintering, or when the fruit are different. Um, but yeah, I would say in general, the bacteria that we are after and we observe in Drosophila melanogaster are pretty consistent with some of the bacteria found in SWD.
Awesome. Cool. I also just thought it was really neat and wanted to make a note of it. So as you well know, we know one another.
Yes.
I study the floral nectar microbiome and how it influences attraction of pollinators and other nectar foragers. And I noticed that you mentioned a few times now that Acetobacter and Gluconobacter were
Yes.
both, associated both with the flies and also foraged fruits.
Hmm.
I just wanted to point out to you that those are also very commonly isolated in floral nectars, perhaps
Yes.
unsurprisingly, sort of simple. You
Yes.
probably already knew that. I'm wondering, do we know that fruit flies feed on floral nectar? Is that potentially an alternative
Hmm.
carbon source for them?
Yeah. So there are at least two reports that I came across that show that they do feed on nectars.
Hmm.
In fact, SWD particularly are much polyphagous and kind of non generalist than some people think. There was a study that looked at um, how SWD feed on non-cultivated crops, and they found they collect over 165 different plant species including flower, nectars, different types of fruits,
Wow.
wild berries, and things like that. More than half of the species end up having SWD adults that came out from it. So they collected over 2 years, and at least half of them, 80 something species, had SWD larvae that emerge into adults. So you can see they're actually a lot more complex in terms of their dietary diversity. They do feed on all kinds of things. And I think microbes plays a role in guiding what are suitable host plants that they can invest in.
Yeah, that's so fascinating. It would be interesting to look at, foraging cues versus oviposition cues on those different
Right,
plant
right.
substrates as well.
Absolutely, absolutely.
All right, I'm going to put out a last call for any questions from our attendees. And other than that, I just want to thank you again, Professor Wong, for giving us a peek into your research. It's a great topic.
Yeah, thank you. It's my pleasure. And if any of the audience interested in my work or have more questions, feel free to contact me by email or even follow my social media.